Good morning, everyone. Today I'm going to talk about AP Biology topics 2.4 and 2.5, dealing with our cell membrane and membrane permeability. First off, topic 2.4, the plasma membranes. Our learning objectives are to describe the roles of each of the components of the cell membrane in maintaining the internal environment of the cell, and describe the fluid mosaic model of cell membranes. So first, here's a basic diagram of a cell membrane. We have two layers of phospholipids, referred to as the phospholipid bilayer. We have a few different protein molecules and a carbohydrate chain. One of our proteins is specified as a protein channel. So our components. We have the phospholipids. These molecules are what make up the bulk of the cell membrane. They are described as being amphipathetic, meaning they have a hydrophilic region and a hydrophobic region. Because they have both of those regions, we're going to see that they only let certain molecules pass through them. And that'll be more important when we get on to membrane permeability and later in our unit as we move on to talking about different types of cell transport. Then our proteins. Remember that proteins are made up of chains of amino acids that have been folded together to form a three-dimensional shape. With our proteins, we've got two broad categories. Those that are integral, so if we look down here, integral proteins pass all the way through the membrane. We have another integral protein here, another integral protein here, another integral protein here, passing all the way through. Peripheral proteins are bound only on one side of the membrane. So here we have a peripheral protein that is bound on the inside of the cell membrane. We could also have peripheral proteins that are bound on the outside of the cell membrane. But notice that this peripheral membrane down here does not go all the way through the membrane. Our different proteins are going to be very important. Again, when we move on to cell transport, this is all building. With our proteins, especially those integral proteins, some of them are going to be channels, which means they're a transport protein. This is going to aid molecules that cannot pass through our phospholipids to pass through our cell membrane. We're going to see that there's several different types of transport proteins. We're going to see that there's protein channels, like in this diagram. We're also going to have protein carriers and protein pumps. Again, we're going to get into more detail on that later set of notes. Then there are also some additional carbohydrates and lipids. We're going to find these on the outside of the cell membrane. So in our picture here, we have got a carbohydrate chain. And then over here we have a glycolipid. So this is a lipid that has some glucose attached to it. Here we even have a glycoprotein. Those, the carbohydrates, the glycolipids, those are going to be used in cell-to-cell recognition. It's going to help cells identify one another. These will be more important when we get on to cell communication. Our cell membrane is often described as being a fluid mosaic model. The fluid portion refers to the weak interactions between the phospholipids that allow for lateral movement, and that's important for the permeability. I like to think of all my little phospholipids. I mean, they even kind of look like people with a head and two legs. In this image, we can see a crowd of people. It looks like one solid crowd, but all the little heads are shifting around. And that's important for permeability, letting things pass through. This flexibility and fluidity can be and is stabilized by cholesterol molecules. You can think of cholesterol, these would be barriers or walls that then the little people in this crowd would have to move around. Cholesterols can be found throughout the membrane. Certain cells will have more cholesterol in them than others. That helps for stabilization. The mosaic part refers to the fact that our cell membrane is made up of lots of different pieces. In art, a mosaic is a picture made up of many different pieces. So here we've got a little picture of some sea turtles made up of a bunch of different pieces. Mosaic model of our membrane is made up of our lipids, that's the primary component, along with the proteins and the carbohydrates. So now let's move on to our membrane permeability, which is topic 2.5. In this, our learning objectives, you should be able to explain how the structure of biological membranes influence selective permeability and describe the role of the cell wall in maintaining cell structure and function. So when it comes to things being selectively permeable, that means only some substances can cross. Some cross more easily than others. Cell membranes are selectively permeable due to their structure, primarily because of those phospholipids. Those phospholipids are going to allow small non-polar molecules, because that's what the bulk of our cell membrane is. Things like carbon dioxide and oxygen pass through easily. They can fit through all those little phospholipids and come through our membrane. Polar molecules like water, ions, and large molecules, such as sugars, do not pass through as easily. Water can kind of squish its way through. Given enough time, some other things might pass, but it's a lot harder. Those are where the transport proteins come in. Some transport proteins are going to allow anything to pass through. Others are going to be specific, and they help regulate the transport of these molecules. There's also a special transport protein called an aquaporin, which becomes very important in allowing water to pass through the membrane. With our cells, there are some additional cell surfaces that allow for permeability and transfer or slow it down. First one is the cell wall. We find cell walls in plant cells, fungus cells, and bacterial cells. Function is all pretty much the same regardless of what they're made of, and you do not need to know all the individual components of the cell membrane. What you need to know is that the cell membrane is there to protect, it helps it maintain its shape, and it prevents excess water loss, or water movement, I should say. It makes it harder for water to leave because now it's got to pass through that cell wall. So here we've got a cell wall of a plant. Here we've got cell walls of some bacteria. In animals, because they don't have that cell wall helping to provide shape, they have an extracellular matrix that's outside of the cell and in between the animal cells. This aids with communication between our cells. And so a lot of those carbohydrates that we saw, some of the carbohydrates, some of the glycoproteins are connected to that. Our cell junctions. These are going to be places where our cell membrane has holes, or excuse me, our cell wall has holes. In the case of the plasmodesmata, it allows for channels in between our plant cell walls, helps to unify the plant, and allows water to move from place to place. Our others, tight junctions, desmosomes, and gap junctions, are going to be unique to plant cells, animal cells. Plasmodesmata, plant. All the others, animals. Let me get that straight. A few different types. We've got the tight junctions. You'll find this in your skin. It's watertight, a very tight protein complex. The desmosomes also act like rivets, pieces of protein. They help to fasten cells together, but allow for a little bit more movement. And then we have the gap junctions. These are going to be channels between animal cells. Very important in communication, we're going to find gap junctions in our within your nervous cells, passing ions going from, say, a nerve cell here to a mesocell or an endocrine cell on the other side. Allows for direct transfer. Again, those three have been animal cells only.